Hi guys, welcome back to my YouTube channel. So today I'll be doing chapter 4.5. It's one of the comment that I read in comment section box in the previous video. So if you guys don't understand anything, you can always comment at the comment section. Maybe I don't reply, but I always read the comment section. So you can just comment the subtopic that you guys don't understand or the subtopic that you guys want to learn from me. It'll be better if it's subtopic lah, because if it's the entire chapter, it's very hard for me to do a video because my video is based on subtopic, right? So it will be better if you comment the subtopic. Okay, so let's get started for today's class, which is nucleic acid. Now a lot of students, they don't understand about nucleic acid. Actually, it's very easy. It's a very easy thing for you to understand as long as you get the concept of it. Nucleic acid, we have a thing called element. Okay. Anything that we learn in chapter 4, we also have element. So for nucleic acid, we have this thing called Chon element, CHON. If you guys don't know what is element, you can watch back my previous video, which is under carbohydrate. Now, when it comes to element, right, the major element is CHON, right? And then we have other element, we have trace element. Nucleic acid contains of 5 different elements, which is C, carbon, hydrogen, oxygen, nitrogen, and phosphorus. So in total is 5. Cannot write CHONP in exam. You have to write carbon, hydrogen, oxygen, nitrogen, and phosphorus in total. Okay, you have to write the full name when you ask about element. Now, after that, we have to learn about a thing called basic unit. Basic unit, it means the smallest molecule. If you guys don't know, please go and watch the carbohydrate video. Okay, you have to watch the video before you watch this video anyways. Basic unit, it means the smallest molecule, right? The smallest molecule for nucleic acid is known as nucleotide. It comes in 3 different shapes. We have the one round in shape. As long as you see a circle, a round shape, it means a group. Okay, this group called phosphate group. Okay, it is known as phosphate group. Next, if you see a 5-corner thing, in maths we call this as a pentagon, right? So in biology we call this as a pentose sugar. Pentagon, right? Pentose sugar, it makes a lot of sense, right? Or they call this as 5-carbon sugar because we have 5 corners. We call it 5-carbon sugar. Whichever one also correct. But I prefer pentose sugar because it's easier to remember. So the round shape and the 5-corner shape is a fixed shape. Okay, it's a fixed shape. And then next one, we have this thing called nitrogenous base. Nitrogenous base can come in many different shapes. It can be something like that. It can be something like that. It can be many different shapes. So as long as they are not round in shape, as long as they are not pentagon shape, then it will be a nitrogenous base. So this is the shape that you have to remember. In exam, they ask you to label. So you need to be able to label them now. Phosphate group, pentose sugar, nitrogenous base. Okay, now done with basic unit, we have to learn what are the type of nucleic acid. We have DNA, we have RNA. I believe you guys heard of DNA because it's the one that contains of genetic information, right? RNA and DNA, they are same but different because DNA, RNA, ribonucleic acid is actually a copy of DNA. So they are same but different kind of thing. Now, before we learn about what is the real difference, we have to learn about the structural difference. It means based on structure, what are the difference that do they have? RNA stands for ribonucleic acid, right? DNA stands for deoxyribonucleic acid. Number 1, the structure of RNA is shorter, okay, shorter. And then the structure of DNA is longer. How do you want to remember? Deoxyribonucleic acid, the name is also longer, right? So quite obvious that the structure also longer. So it's very easy for you to get the idea, right? Deoxyribonucleic acid, the name is longer, and then the structure is also longer. Compared to RNA, like it's also longer. Second, ribonucleic acid contains of ribose sugar, ribose called ribonucleic acid, right? Ribose sugar. Deoxyribonucleic acid contains of deoxyribose sugar. Okay, it makes a lot of sense because the names are quite similar. Ribonucleic acid, ribose sugar. Deoxyribonucleic acid, deoxyribose sugar. Okay, this is the other structure difference. Now, third difference is the strand. RNA is only single, okay, only one single. DNA is double-stranded. Just remember, deoxyribonucleic acid, D, remember double. Now, what does double-stranded means? It means they have 2, something like that, okay, double-stranded. But RNA, there's only one thing. Now, because they are DNA, deoxyribonucleic acid, they are together, but RNA is forever alone. So this deoxyribonucleic acid, right, they have a pair, means got a pair that can hold hands. Anything that can hold hands, they can form a bond, right? They must have a bond between them. They can hold hands, it means they have a bond. the bond between them is known as hydrogen bond. Okay, the bond between them is called hydrogen bond. Now we have to learn how they hold hand, or we have to learn who they hold hand with later on. But just now we are just learning the structural difference. Okay, so these are the three structural difference. Number one is the size. Which one is longer, which one is shorter? When the name is longer, it means the structure is also longer. Second, the sugar. If it's deoxyribose sugar, it means they are under deoxyribonucleic acid. Third one is the shape. So if it's ribonucleic acid, it's single, forever alone. If it's deoxyribonucleic acid, it means double. Double means they have a pair. If they want to have a pair, they must have a bond called hydrogen bond. Okay, now what is the difference between DNA and RNA? The difference, the real difference between this one is the genetic information. DNA, they have the gene of CGAT. Can you see CGAT, which is known as cytosine, guanine, adenine, and thymine? And then for RNA is CGAU. Okay. The only difference is U and T, right? So in exam, if they ask about differences, you can write uracil as differences and thymine as differences. Similarity is CGA. Because RNA got no pair, actually they have a pair, just like we don't learn in biology. They have a pair, or we just don't learn in biology. So we learn the pair in DNA. Now, what is the pair? Who do they pair with? C always pair with G. Of course, G always pair with C, right? Obviously, because it's only have two. If C is G, of course, G is also with C, right? And then A, they will pair with T. T, we will pair with A. How do you want to remember? Remember as at GC. You always use Google Classroom, right? At GC. A with T, G with C. Then obviously, you know that T is with A, C is with G, right? At GC. This is how you remember the pair at GC. So you look at the diagram, right? You can see that G always pair with C. C always pair with G. T always pair with A. A always pair with T. So T cannot pair with C, cannot pair with G. Remember at GC, ATGC, because they will always ask you to write the combination, or they ask you to draw the pair out. If the question asks you to draw the pair out, how are you supposed to draw? They give you one side. Okay, maybe they give you this side, okay? Because this thing called nucleotides, right? They ask you to fill in the DNA. Because if it's only one side, it means it's just RNA. They ask you to draw DNA. How are you supposed to draw DNA? Number one, you have to draw the bond. The bond name called hydrogen bond, right? We have mentioned that hydrogen bond is the whole hand one. And when you draw the nitrogenous base is the one that contains of genetic information. So the nitrogenous base can be A, can be T, can be G, can be C, can be anything, depending on what they give it to you. And when you draw, you have to draw according to the shape. So if this is like a sharp area, and the shape that they should buy with the sharp area is fit one, like lock and key hypothesis. Okay, they must be fit. Cannot be like random shape. It must be a correct shape, okay, in order for them to form a bond. So this is the bond in between, and you have to draw the thing. And the one that you have to take note is the one you draw it on the other side is inverted. Everything is inverted. So this is like that, right? The other side is inverted. If it's not inverted, your answer is wrong. So remember, whenever you draw a pair for DNA, everything must be inverted. And the pair shape, it must be correct. So this is something that you have to take note for DNA. For drawing part, remember it's inverted when you draw. Because a lot of students, when they draw, they didn't draw the inverted diagram. If it's not inverted, your answer is wrong already. So it must be inverted. Okay, so remember at GC, AT and GC. Next, besides the genetic information, the nitrogenous base is different. The function of DNA and RNA is different. The function of DNA is to synthesize protein, is to produce protein. If they are used to produce protein, they can find in ribosome, cytoplasm, and nucleus. Nucleus confirm got DNA and RNA, because nucleus is the one that contains of genetic information, right? Everything starts from nucleus. Nucleus control all cell activity. So ribosome is the one that synthesize protein. Of course, it can be found in ribosome as well. Cytoplasm is the space, okay, the biochemical reaction area where it takes place, cytoplasm. So for DNA, what is the function? They will carry genetic information. If they are used to carry genetic information, where can they be found? It can be found in nucleus, in plant cell, they can be found in chloroplast and also mitochondria. These are the part that you have to know, DNA and RNA, because in exam, they ask you to write differences and similarity. So you have to know what is the similarity, you have to know what is the differences. Okay, now for those who don't understand what is a polynucleotide, poly means many, a lot, called many. Nucleotides, it means this one is called nucleotides, right? So they are formed by many nucleotides. We call this as a polynucleotides. So how does this DNA, how does this RNA is being formed? It's being formed like that, okay? We have 4 sentences over here. I'm going to use 4 words for you to understand these 4 sentences. So first of all, everything starts from DNA, right? DNA. So DNA, they will join with a thing called histone. Histone. Okay, you can see there's a word called histone here. If you look at this diagram, you can see there's a word called histone here as well. This is histone. It's like small little thing here that join them together. So they will combine with histone to form into a thing called nucleosome. Nucleosome can be found in, it can be found in the nucleus that I call nucleosome. So this nucleosome, they will intertwine to form into a chromosome. What does intertwine means? Intertwine means like that. Okay, they will wind up, called intertwine. Because chromosome, right? How do they look like? Chromosome looks something like that, like a butterfly shape. So if you look it under the microscope, you can actually see something like that. This is the chromosome. So this is how they actually form a chromosome out. Using the 4 sentences, using forward, you can understand lah. Okay, DNA, they combine, they join with, wounded around with the histone, they will form into a nucleosome. Nucleosome, they will intertwine to form into chromosome. So this is how we actually form, or how do we actually have the chromosome? How do we actually contains of carries genetic information? Everything started with this thing. Okay, so it's a very short subtopic. If there's anything that you don't understand, you can always comment at the comment section below, or any video that you want to watch, you can also comment below. So I will see you guys next time. I don't know what chapter that I'll be doing, but see you guys next time. Hope you guys understand the video. Okay, bye guys. Bye.